Our last presentation before the break is leveraging host gene expression to diagnose SARS-CoV-2. Will be presented by Dr. Ricardo Henao, Assistant Professor of Biostatistics and Bioinformatics, well as Electrical and Computer Engineering, and member of Duke Center for Applied Genomics, Precision Medicine, DCRI, and Duke AI Health. Ricardo?
Thanks, Michael. Can you see my slides?
Yes.
Okay. So to begin with, I'd like to mention that this is a very small piece of a very large effort from the Duke Center for Applied Genomics and Precision Medicine. Specifically, it's infectious disease arm that has been working for about 10 years or so on biomarker discovery for infectious disease and translation of some of those technologies to practice. This is a proof of concept study for investigating the ability of host gene response to identify COVID infections. So a little bit of a bottom line of what I am going to show you today is that based on gene expression and a cohort that has been collected over the last 3 months or so, we have been able to identify gene expression signatures that show a robust host response that is partially consistent with acute respiratory infections. The keyword here is partially, because that signature being partially consistent is what is going to be able to make it sensitive enough and specific enough to flag COVID-infected patients from other respiratory infections and healthy controls. We will show that these gene signatures can accurately identify COVID patients from a variety of controls, which I will introduce in a minute, and that the accuracy of the model based on gene expression is preserved over the prolonged course of the COVID-19 infection and across a wide variety of ranges of symptoms and symptoms severity. Now, before I show you some of the results, let me just tell you a little bit about the cohort that we are using for this study. So we are using 46 PCR-proven symptomatic SARS-CoV-2 infected subjects, 14 of which have been sampled at multiple time points. And the time from symptom onset ranges from 1 to 35 days. We have divided these time curves into 3 buckets: early, mid, and late, where early is defined as the first 10 days since symptom onset, and late as more than 28 days since symptom onset. As comparators, we are using 19 healthy controls and a panel of 59 seasonal coronavirus collected in previous years, 17 influenza patients, confirmed influenza patients, and 20 bacterial pneumonia patients, all that can be grouped as acute respiratory infections. Now, as for the first conclusion of the study, that SARS-CoV-2 triggers a robust host response, what you can see on the figure on the left is that there is a large collection of genes from which I am only showing the top 100 that shows very marked differences between COVID-19 patients at all time points and all the other comparators, including healthy controls, bacterial infections, and other viral infections. But in addition to that, that host response, that some of which characteristics are manifested as upregulated interferon signals, an example of which you can see on the right, are very characteristic of respiratory infections. And what I mean by that is that early on, they look like common respiratory infection like influenza, but as time moves on, it resolves and starts looking more like a healthy characterization for patients. Now, this ties back to what I was saying before about the partially consistent with respiratory infections, because only interference signals are characteristic of that response, but as I will show you next, we can find genes that are more robust across all time points. So on that, we just built a model on this data, and we can show that the model is highly accurate. So it's both sensitive and specific at identified COVID-19 patients from healthy controls, seasonal corona infections, influenza, and bacterial. On the left-hand side, you can see AUCs from receiver operating characteristics showing very good performance characteristics. And just to show you how that looks in terms of the predictive ability of the model, on the right, we show you those probabilities for COVID-19 patients, influenza patients, and seasonal corona patients, where you can see that the model is very accurate with the probabilities that are being predicted for these subsets. Now, most importantly, if you look at the figure on the top right, where we have stratified COVID patients into early, middle, and late symptoms, you can see that the model sustains its high confidence predictions across all time points, which is very important if we want a robust model to identify COVID patients. Now, where this ties back to practice is that if we look at symptoms over time, which are shown on the bottom left of the slide, you can see that only at presentation, symptoms are very high. The figure shows symptom severity, but for most of the time points for which we are obtaining and classified patients or COVID, symptoms are actually very low. And for most of those data points, PCR tests are likely to result in negative results. However, the prediction from the model are, again, highly confident, which give us hope that this is going to be useful for identifying early COVID infections. In addition to that, we also stratify by hospitalized subjects versus not in the middle panel, labeled as treated at a hospital, where we can see very conserved and very marked differences in those patients that are more severe infections that does need to be treated in a hospital, that are also captured by these signatures. Now, as a conclusion, this expression from a small set of genes allow for the detection of SARS-CoV-2 patients, as well as being sensitive and specific in the context of other infections, including bacterial and other viral infections. Now, this model, based on these genes, combined with rapid nucleic acid platforms, will allow for translation, so they can thus be used for early detection of COVID-19. As a caveat, I will say that early detection needs to be properly validated on exposed but asymptomatic subjects, which we are in the process of acquire an assay. But what I would tell you is that we have work on influenza, where we can show that similar gene signatures can identify early influenza infections, and this is from a paper that is going to come out soon in Lancet Infectious Disease, which again, combined with the results that I showed you before, gives us hope that these genes in combination are going to be able to detect COVID-19 infections before the symptoms manifest. Thanks.